So at ASCO GU 2025, we presented our findings from a second revision analysis evaluating the efficacy of lutetium PSMA with or without ARPIs or androgen receptor pathway inhibitors for the treatment of metastatic castration-resistant prostate cancer. Speaking of Vision trial, it was a phase 3 trial, multinational, evaluated the efficacy and safety of lutetium PSMA with standard care versus standard care alone in treatment of metastatic castration-resistant prostate cancer. For that trial, the eligible patients were the ones who had metastatic castration-resistant prostate cancer with at least one prior RP, a androgen receptor pathway inhibitor, and one or two taxane-based chemotherapy. And they found that there was a improved PFS and OS for the patients who received lutetium PSMA, which led to the approval of FDA for treatment of patients with metastatic castration-resistant prostate cancer. But in this secondary analysis, we specifically focus on the patients who only received lutetium PSMA. And our key question was, how would be the efficacy and safety of concurrent ARPIs with lutetium PSMA for treatment of these patients? So we had two arms, the ones who had lutetium PSMA with concurrent RP versus the arm for the patients who received lutetium PSMA without concurrent RPs. So we retrospectively analyzed the efficacy and safety profile. First of all, speaking of the patient characteristics, as the trial was not designed to look at these specific questions, although most of the characteristics were balanced between two arms, including median age, advanced Gleason score 8 to 10, and metastatic site distribution, but there were some imbalances like PSA level, LDH level, and more prior taxane-based chemotherapy in the patients who did not receive RP. Taking that into consideration, there was 5.5 OS benefit for the patient who received concurrent RPs, 17.8 months for the patient who received concurrent RP with lutetium PSMA versus 12.4 months for the patients who did not receive RP with lutetium PSMA. However, the PSA progression-free survival and radiographic progression survival was not statistically significant. Both of these endpoints were trending toward better outcomes with concurrent RPs. PSA PFS 8.6 versus 7.3, and for radiographic PFS, it was 10.2 versus 8.5 months in favor of concurrent RP with lutetium PSMA, but neither was statistically significant. Coming into the safety profile, which is super important, the overall adverse events rates were similar between two groups. There was no new safety signal in the patients who received concurrent RPs, and it was well tolerated, manageable for those patients. And death due to adverse events were reported in 8 patients, 2.8% of patients with concurrent use of RPs, and 4.5% in patients without concurrent RPs. However, we need to acknowledge the limitation of study, the retrospective nature of analysis can introduce some unmeasured confounders affecting the OS results, and multi-variable analysis is also needed, which is already performed and will be reported in our manuscript. In terms of the findings that we had in our study, it is actually explainable by the mechanism of the androgen receptor pathway inhibitor and PSMA lutetium. By blocking the androgen pathway, it can lead to the upregulation of PSMA on cancer cells, which can lead to the improved benefit from PSMA lutetium. Our study findings actually were aligned with the ENZA-P study that was presented at ASCO GU, which showed combination of enzalutamide and lutetium PSMA led to improvement of PSA and radiographic progression-free survivals, and improved overall survival with around 8 months. But however, because of the confounders in our study and retrospective nature of analysis, this study was more determined to be thought-provoking and hypothesis-generating for the future trials. We will be conducting randomized trials to evaluate the efficacy and safety of concurrent ARPI with lutetium PSMA for treatment of patients with metastatic castration-resistant prostate cancer, to evaluate the findings that we had in this secondary analysis.